So, to connect with the previous lecture, we were talking about various approaches to cultivate unculturable bacteria, right? Considering their importance in the nature, in the ecosystem, and sometimes these are the unexplored treasure that would provide us with some novel drug molecules or industrially important components. So we need to tap this diversity. And those which are not growing under artificial laboratory conditions are to be explored, are to be grown under laboratory conditions. Various approaches to facilitate the growth of these once unculturable bacteria is a challenge, but various approaches to that we are discussing. Now, when it comes to different approaches for separating and growing uncultured bacteria, the scientists are making various efforts to improvise the developed methods as well as to find the new methods. So we have talked about the diffusion chamber, the trap, right? Mimicking the mimicking the natural environment in the laboratory. So the next method, which was developed, I think, in the early mm, 21st century, that is 2001 2002, by Kaler et al. It is talking about encapsulating single bacterial cell in the micro droplet, and the droplet is made up of some gelling material, maybe say agarose, right? So like solidifying gels, you explore for entrapment of enzymes and yeast cells, etc. For industrial microbiology, you have learned that. So maybe agarose you can use, maybe alginate you can use, or other natural polymers, they can be explored. So these people, they had talked about use of agarose micro droplets in which a single cell was entrapped, right? So how it is possible to entrap a single cell? So the experiment included collecting the sample from the environment and going for a dilution, and dilution to an extent that it is going to like limit presence of bacteria. That is highest dilution you are going to go with, extensive series you are going to go with, all right? And that dilution you are going to use to put into gel form. So this, I mean, that diluted sample you are going to use to prepare the gel beads or the droplets, right? So gel micro droplets, short form, if you go for it, is accepted as GMD, all in capitals. So you are going to encapsulate the cell in a gelling matrix like agarose, and you are going to fill it in the column, and the column is going to have the beads, each bead with a bacterial cell, and that stack you are going to continuously bath with the natural sample. So for example, it is isolated from marine water, you are going to pass down the seawater down the column, right? Now here, of course, you have to take care that seawater again is going to be the one which should not contaminate your the desired organism, the gel bits, right? So, of course, the flow column, which is bounded by membranes, that is going to be used, right? So, marine samples, when they are cultured, seawater can be constantly flushed through the column, and this seawater only is going to act as a growth medium, and you are going to allow these bacteria to get incubated within that gel beads only. The GMDs are going to remain in the column. The column is washed through filter seawater, of course membrane filters I'm talking about, and then the bacteria may find it good as a nutrient medium, they will grow, and then at a particular time you take that GMD, that is gel bead, out and pass it through the flow cytometer. So if there are any microcolonies present, they will be sorted, right? And you can collect those beads in which colonies are seen or presence of bacterial cell is seen, and then that bead you are going to use as a starter inoculum. So may you put it in the liquid media, or you may put it in the microtiter plate, which are going to be used as culture plates, etc. Or maybe directly you are taking it for sequencing and say phylogenetic analysis, right? Of course you can use it for further cultivation as well as for DNA sequencing, etc., etc. So when you are using the real seawater as a medium, very high number of bacteria could be isolated. You can explore the higher diversity from the water rather than the added nutrients, right? So how it was determined if which medium is good. So both the media, you are going to have two different strands, beads filled in both the columns. Nutrient medium along with the seawater is passed down one column, and second column is bathed with natural unadulterated, or I should say unsupplemented plain seawater. And then whatever the gel beads are showing microcolonies, they are taken ahead for sequencing and find out the bacterial diversity. So when you talk about these GMDs, you can get a diverse type of bacteria which are trapped and again as pure colonies because they are developing from a single cell. Till now, whatever methods we talked, there was a challenge how to purify them. Now, here this question is sorted out by implementing a kind of a what you can say, chamber, right? And each micro chamber is entrapping one cell. So, whatever microcolony develops within that chamber is going to be a pure colony of that isolate, right? And yet it is in spite that it is in this it is in the culture medium which is very much similar to its natural environmental conditions. It is still a separate and pure colony because it is entrapped in a gel bead and has emerged from a single cell. So uh, for example, this GMD has given what you can say uh, 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 mm, the most uncultured ones, bacteria, that is Plactinomycetes group, right? They were previously uncultured, but because of the GMD technique, more than 84% of of any published 16S sequences, uh, they were grown or cultured. So this included the sequencing that was done, of course, of the original seawater sample first, and that indicated the possibility of presence of this organism. And then this enriched medium and the isolates obtained, they were again 16S sequencing was done of those, and it was concluded that they belong to Plactinomycetes group. Now here, agarospears can be used to encapsulate, or maybe polysulfone is another material which is used to encapsulate and incubate bacteria. Now, here we are talking about a column, right, in which the beads are kept. But a step ahead was done by Kushmaro et al., who had done a wonderful job that they had used the agarose spears, and they had directly inoculated them onto the corals, right? Natural corals. So incubated those bacteria uh, 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 yeah, in association with the mucus, which is the natural source of diverse nutrients, and it is there in the native environment of those bacteria. So, on the surface of live corals, they had put these gel beads and have seen whether any specific bacteria are growing and are we getting any new isolates from this. So, such diverse, interesting approaches are used by scientists to grow the uncultured bacteria. And now it is really very required to determine what particular aspect of the environment is important for the growth of that bacteria rather than just putting the entire environment, right? So, in this case, uh, you have to identify the desired nutrient or desired growth factor that is demanded by bacteria. So, here I'm sharing the slide which talks about GMD, but is that just enough to go with GMD? Fine, GMD will provide it the nutrients which are naturally available. That is true, because you are bathing it in the soil extract or the freshwater sample from where you have isolated the bacteria, or seawater directly you are passing down the column. But out of this, if you are able to find out what actual nutrient is the demand of the bacteria, then it would be easy to propagate it under lab conditions and scale it up, right? So the approach here, which I'm talking about, is to identify what particular aspect or what particular growth factor is essential. So in this case, one example is that of, say, you have the leeches. Have you heard the blood sucking organism, right? So the gut flora of these organism, if you want to isolate, how would you go? All right. So here, uh, the examples of Rickenella like bacterium, which is found in the leech gut, those were isolated. These are again an example of an environment which is having a diverse microbial flora, and many remained uncultured for a long time. But this story talks about use of the mucin, which is there in the gut of this leech, that is as the carbon and energy source for the once uncultured Rickenella like bacteria. So the environment that is important for their growth, but what specific component of the environment would help the growth, that needs to be identified. So high throughput sequencing, that is by molecular techniques we are talking about, the RNA transcript studies were done to determine that uncultured Rickenella like bacteria in the leech gut, and that bacteria is present in the naturally in the gut of the organism, but how to grow it? So they had prepared the media containing the mucin, and yes, they could find this Rickenella growing onto that medium. All right. Now when it comes to GMDs, you can of course put these GMDs in media. I mean, you can mix these two approaches. That is, a selective component added to the medium, and then you are exposing the GMDs to that. All right. So selectivity is there, yet a single isolate would be growing within a microbead, so it will remain as good as a single isolated pure colony. All right. So when you're talking about this GMD, these are the steps for your understanding. I had just rushed through while reading it. You can just see what are the steps. You can go for single cell encapsulation. For single cell encapsulation, what you need to do is a series of dilutions, and then the particular unit volume, say per ml, you should have only 10 cells. So per 100 microliter, per 10 microliter, probably if you have one single cell, that you are going to put into the gel bead. Like that, you can, of course, these are automated systems, and you can entrap single cell and prepare the beads by automated bead preparing systems, right? So encapsulation can be done by microfluidic devices, and small fine micro droplets can be prepared, which are going to capture the bacteria of your interest. All right. Now, of your interest, that means you have to prepare it for all the bacteria which are present in it. And so many, if for example in a given sample there are 10 different species, you will have uh, at least 5, 10, 15, 20, 100, whatever the number of cells per unit volume would be of the particular species. And each bacteria you are preparing the microdroplet of, and then it will be passing through, and then each of these bead you are going to study further. May it be sequencing, may it be phylogenetic studies ahead, etc. Right. And then my bacteria of your interest can be traced back. Now high throughput methods, like that is what molecular techniques what I'm talking about, that you can use to find out the bacteria, the one which was unculturable earlier but is now been detected in the sample. So selective enrichment of these GMDs you can do, and flow cytometry will help you to find out the beads which are having the microcolonies, and then you can go for rapid detection and enumeration methods, and use each single bead as a starting culture in the new plate for preparing the pure culture of that isolate, right? So you can blend the two techniques, like uh, entrapped bead where a specific single isolate is there, and you are putting it into the medium which is having a selective nutrient like mucin from the leech gut was put into the media, and it could grow very happily in that, right? Rickenella bacteria could grow in that. So similarly, any uh, growth factor which is essential, you can find out. It can be siderophore, right? It can be catalase required to reduce the oxidative stress. It can be growth factor provider like mucin, which is a carbon and energy source, anything. But you have to identify that and provide it in the medium. And to isolate the organism in a pure form, you can entrap a single cell in the agarose or similar synthetic or natural polymers and put it onto the growth media or in the growth media if it is a liquid culture medium. All right. So what different kinds of requirements you may come across when you are talking about unculturable bacteria, right? When it comes to unculturable bacteria, siderophores is the one which we talked about. The example of siderophore, E. coli, enterobactin, and Maribacterium example we had talked about. Then we talked about catalase from Prochlorococcus and Alteromonas. Then common nutrients. It could be some specific growth factors, or maybe specific stress factors. You have to find out what is the hindering agent or what is the promoting agent, and then you have to provide the promoting agent, or you have to remove the hindering agent so that you are able to promote the growth of that bacterial species. All right. Now, when it comes to growth of unculturable bacteria, there is another aspect, host assisted environments. So that is what example of co-culture with lead gut mucin we talked about. All right. So co-culture dependence and environmental incubation requirements, if you talk about, of course, always not necessarily to be focused on host associated environment. It depends on what kind of bacteria it is. All right. So bacteria from human microbiome also people are trying to explore, because your microbiome is going to decide your health condition, or it is going to cause disturbances to your health, because bacterial health, microbial health of your gut and your own body health, they are very much intensely connected. So you will wonder to know simple things like very thin people or very fat people, they have a distinct microflora. Similarly, the sweat of the body, the metabolic pathways, and so many things, the common natural normal flora inside and outside is going to regulate and contribute to your metabolic processes and overall health. So people are looking about for human microbiome, which are very important in health and diseases. So the potential role of these host associated unculturable bacteria is also one of the areas where the application of unculturable bacteria and the essence or importance of studying these uncultured bacteria is there. It is to be investigated, right? So when you're talking about an attempt to isolate, that is one thing, but you need to quantify, right? When it comes to examples like unculturable for human health and disease, so maybe the bacteria is present but number is not adequate, right? So it will be a kind of a competition between the normal flora and the opportunistic pathogen from the environment, and if the normal flora is good enough and strong enough, your health is maintained. So the quantification is very important, right? So, in an attempt to quantify the role of the number of these organisms, you have n number of instruments, but at the same time, you need to assure that this particular bacteria has this particular role. So, you have to quantify or identify or know the role of culturable and unculturable members, you can say, of the gut microbiome. All right. So, for that purpose, you will have to take help of some experimental animals. So, these unculturable bacteria can be isolated by various techniques that we talked till now, and they can be inoculated into germ-free mice as host, and then will be like transplanted into human intestinal microbial communities. You want to check that. So the experimental animal is mice, right, as a host. And then the fecal samples from the donors are directly inoculated into germ-free mice. As an example of what is the source, of course, the fecal one. If we are talking about gut microflora, right? So the fecal sample bacteria are isolated from that, and they are inoculated into germ-free mice. So, uh, or maybe you are going to put them on Petri plate for culturing. But if they are not culturable, then you have to go with introduction into mice. So there are n number of strategies and experiments and methods to make these unculturable bacteria grow and use and so that we can use them. All right. So when, for example, if you're talking about the mice, the germ-free mice inoculated with bacteria isolated from human fecal sample, these bacteria will start growing there and the mice will get colonized with the bacteria, right? And here the cultivation step, which is like equivalent to your inoculating the mice, there are different ways to inoculate, right? So you can simply inoculate it through oral route, or you can put it into foot pad, right? Whenever you want to see the effect of particular bacteria. So directly you can colonize these germ-free mines, and they can host the bacteria that you want to study and find its role in the metabolism of human systems, right? So microbiome, which respond to dietary perturbations, now there you have to have that it should be fed to bacteria, the bacteria should be fed to the mice, or it should be implanted into its intestine, right? So direct transfer of the bacteria is very important. So how it can be done? Is it by injection way, or it is just like injection in the sense not intravenous or intramuscular, that the syringe is filled with the suspension and it is just put into the mouth of the animal as like a feed, uh, like feed pipe. You are going to put that bacteria into its system. So there are different ways what people are attempting to cultivate these once uncultured bacteria, and you have to find out the exact requirement of bacteria and then make them cultivable. All right. So with this maximum number of approaches for growing the unculturables that we have talked about, right? If you have any query, you can ask, or else we can end today's lecture over here.